I wanted to take a few minutes and explain some of the labs that you're going to be doing with photosynthesis. First thing you're going to do is a chromatography lab where you're going to take a strip of chromatography paper and you're going to roll like a quarter over a spinach leaf on top of the paper, probably about an inch up, and it's going to leave pigments from the spinach on the paper. The chromatography paper then gets placed in a long thin tube, sort of like a test tube. I have some little chambers with an alcohol at the bottom that helps move the pigment molecules up the paper. And just like in gel electrophoresis, things move according to their size. So you should actually be able to separate the pigments that are found in the spinach leaf using, they'll bind to the alcohol molecules and they'll move up. The lighter pigments will be able to move the furthest on the chromatography paper. The most dense, heavy pigments won't get as far. So you will see separation, and hopefully you'll find chlorophyll A and chlorophyll B and maybe some carotenoids and maybe some xanthophylls. So that's one thing you're going to do. Another experiment that you're going to run, I'm going to give you choices. You can decide what you'd like to do. Here's an example. I have red cups and green cups. And you are going to first run the experiment as a control with just a clear cup. And what you do is you take spinach leaves and you use a hole punch. And you punch out circles, which makes them all the exact same size, which works out fabulously because we can calculate the area of the leaf that we're using quite easily. And we can make sure that we have a constant amount in each one. So we're going to punch out these leaves. And when you punch out leaves, the next thing you do, you want to load them with carbon dioxide. I mean, give them as much carbon dioxide as possible so that they will be able to do photosynthesis. So you're going to actually take a syringe, open it up, put the chloroplast down inside of here, hold your finger on it, add some bicarbonate solution. Bicarbonate solution is baking soda and it has a lot of carbon dioxide gas in it. Put this back on, push it down, and then with your finger go back and forth like this. And as you do this, the spinach leaves will take in enough bicarbonate that they will sink. They will actually have so much of the bicarbonate inside them that they become more dense than water, and so instead of floating like they normally would, they will sink. Once they're sinking, still hold your finger on the end, open this up and dump it into your clear cup. When you put it in the clear cup, you are going to have your cup under a certain condition. So I want you to have one just in the room, one in bright light, and one in the dark. And I want you to, every minute, I don't know, maybe I'll change it as we go through, but every minute count how many discs, so discs of spinach, are floating. Now, what would cause them to float? As they do photosynthesis, they're going to use up the bicarbonate, so using up the carbon dioxide, and they're going to produce oxygen gas. As they do that, using up the bicarbonate and releasing the oxygen, they should become less dense and be able to float. So over time, you're actually going to be able to calculate the rate of photosynthesis by the amount of floating discs per minute. Under different conditions, hopefully we should be able to see the effect that those conditions have on the rates of photosynthesis. Then I want you to design one yourself. You could try different colors of light. Green light only coming through this, red light coming through this. If we have some other cups, we can do some other colors. You can add different substances. You can make it cold, you can make it warm, you can make it acid, you can make it base. You decide what cool things you want to do and try it out. Now, the third experiment that we're doing, you're going to take spinach leaves again, so that's going to be our plant of choice. You can tell by looking at spinach it has a lot of chloroplast per cell. So you're going to take a chloroplast suspension, which means you grind up the cells until you have a nice chloroplast suspension. When you grind it up, you're only breaking up that spinach so that we can use it in these little cuvettes, these little tubes. You're not actually, excuse me, you're not actually destroying the membrane of the grana, not the thylakoid membrane at all. So if it's intact, it should still be able to do photosynthesis just fine. Of course, you need the membrane to be intact, so we aren't, destroying the cells entirely. We just want to be able to get it into a solution. So once we have a solution of chloroplasts, we are going to take and put them in these little cuvettes. And again, we want to measure the rate of photosynthesis. But this time, I have a very large spectrophotometer, or we have these small specs which are called colorimeters. Both of them do the same thing. They can measure either the absorbance of light, so how much is being absorbed by it, or the transmittance, how much is going through. And instead of NADP+, we're going to replace that with a blue dye called DPIP. Diphosphoenol pyruvate. So DPIP is a blue dye that is easily reduced in photosynthesis. When it gets reduced, it goes from blue to colorless. So if we take our chloroplast suspension and we add DPIP to it, it should look blue in here or blue-green in here. When I put it into this colorimeter, it should show me that the transmittance is very low, meaning not a lot of light goes through, or the absorbance is very high. It's really absorbing a lot of light. If I keep this under a bright light, this should start doing photosynthesis. The more photosynthesis it does, the more DPIP it will reduce, changing this from blue to colorless. So if we keep measuring either transmittance or absorbance, whichever one you choose, really both of them are going to show your photosynthetic rate. One is just going to go up and one is going to go down. You will keep measuring that value until you can plot it on a graph and get a rate of photosynthesis. Again, you can do one as a controlled experiment and then change it up. Add different stuff, make it cold, add an acid, add different colors of light, get a black light, try something different and see what happens. Oh, for these labs, you're going to be doing a Prezi. I have a sheet that sort of outlines what I expect from the Prezi. I'm going to teach you how to use a Prezi, create a Prezi. It's a pretty cool presentation tool. I will also show you how you can create a Prezi with multiple users, where multiple users can edit and work on the Prezi. So you can't have that excuse, I did the whole thing myself, or I can't get together with my teammates. You will each be able to access the Prezi on your own with, it's not very easy to do it on a cell phone, but with your own laptops or computers or whatever you're going to work on, you can access it, be working on one part of it while somebody else is working on another part of the Prezi. All right.